welcome family. Before us, we have a question in biology and genetics, and we are looking at sex-linked diseases called the haemophilia. So the question reads, figure 5.1 below shows pedigree diagrams for two families A and B, outlining inheritance of a sex-linked disease called the haemophilia. Now there are other examples of these sex-linked diseases such as the sickle cell anemia, then red-green color blindness, even albinism is also part of this. Now, if we look at haemophilia as a condition, we are looking at a condition in which the blood clotting is affected. So now we have two families. Family A with these two parents, then this is our family B. Then we have the offsprings for each family. Now we can answer the first question, because the first question is all about identifying the family that has a carrier. So question A1, from figure 5.1, which family has a parent who is a carrier for haemophilia? Now, for this condition of a carrier, you can't tell by looking at the shapes, because for a carrier, the circle is unshaded. Then for a normal woman, the circle is also unshaded. So you can't tell by looking at, is this person a carrier or is this person 100% okay or carrier? So you can't tell by looking at these two. Now, what can help you to identify the circle, the parent who is a carrier for that condition? So what you need to do is you need to focus on the offspring. So from the offsprings of family A, we can see that the son, there is one child who is a son who is sick. Then if you look at the children of family B, we can see that all four children were 100%, or let's not say 100%, but we can say they were okay. Now, since family A has a child who is haemophilic, then this family is the one with a parent who is a carrier. So that's what we are going to say. So the answer here is family A. Then the reason we'll say because they had a son who was haemophiliac. Alright, we move on to question B1. Using the symbols X, capital letter H. So whenever you see a capital letter for the question like this one, it means that this chromosome is not carrying a condition or a problem. So the genes that are found here are okay. Then for a small letter, it means that that's where the condition is. Now state the genotypes for offsprings H and J if J is a carrier for haemophilia. Now before we answer this question, let's just take some notes. If possible, I think you can write these points down. So the first one, I'm going to look at a man. So with a man, we only have two conditions. The first one, we have a normal man and a man who is sick. So for a man, there is nothing like a carrier, because the Y chromosome of a man does not contain these characteristics. Now, in terms of the chromosome, the man has XY. So if the man is sick or okay, the only letter that you are going to associate with H is letter X. So for a normal man, you put capital letter H here. So this is a normal man. So normal, then the box must not be shaded. So whenever you see a box that is not shaded, it simply means that the man is okay. Then the case where the man is sick, you need to have a small letter H. So this man is sick. Then in terms of identifying using the shapes, this must be shaded. So a shaded box represents a man who is sick. Away from this one, let's have a look at a woman. So for a woman, we are expected to have 3 conditions. The first one, I'm going to look at, I'm going to put 100% normal. So if a woman is 100% normal, and remember this must be XX. So how should we write these hemophilic conditions? So you put capital letter H, even here capital letter H. So whenever you have 2 capital letters H, then the woman is 100%. Okay. Now what of one is a capital letter, then the other one is a small letter? So what does this mean? So this now means that this woman is normal, but is carrying that defect. So has a problem that can be shown in her offspring. Now let's look at where we only have 2 small letters. So this now simply means that this woman is sick. So a normal woman will have a circle which is unshaded, a carrier will have a circle which is unshaded, then a patient will have a circle which is shaded. So between the carrier and the normal, you can't tell if the woman is a carrier 100%, okay, based on these structures. So what you can use is the offsprings that will now help you to know if the parent is a carrier. So we go back now to this question. So we are told to set the genotypes for offspring H. So let's go back to this diagram. So if you look at H is associated with this box that is shaded, and according to our key, this simply means that the man is sick, and we said if the man is sick, we are supposed to use this one. So this is the genotype that we are going to write here. So X, Y, then the small letter H. Then for J we are told is a carrier. So what are we supposed to do for a carrier? We know that a carrier is represented by 1 small letter and a capital letter. So you put XX, H, then here. So that's what you are supposed to do. Now we move on to Roman number 2. Roman number 2, we are told to, using a genetic diagram, show whether the offspring would be hemophilic or normal when H and J are crossed. So I'm going to write the man here, then the woman here. So I'm going to begin with parental phenotype. So parental phenotype is the parental outward expression. Now, how do you write the phenotype in terms of sex-linked diseases? So for a man, we say this is a patient, so we'll just say hemophilic. So you just have to set the condition. Then for a woman, we know that this is a carrier. So that's what you're supposed to do. Then after parental phenotype, you move on to parental genotype, where we look at the type of genes. Now, we are lucky that we are looking at H and J. We already have the genotype, so we're just going to write them down here. So for the man, we are told he's sick. Then this is a carrier. So you put 1 capital letter and 1 small letter. Then after that, we move on to parental gametes. If you look at the gametes, the simple definition that we can give to the gametes are sex cells. So what are we supposed to do? We need to now separate these. So you write X, Y, X here, the other X here. If you want, you can put them in circles. So these are our gametes. And remember, these are just chances. We are just looking at the chances. Then you can draw your genetic diagram. So this will be XX, capital letter H, small letter h. I'm going to begin with this capital letter H. Then for this one, we have X, small letter h, X, small letter h, because we have two small letter h. Then we move on to this one, we have XY with capital letter H. Then here we have XHY. So we'll call these as the genotypes of F1. So we call these as now the offspring, and if you want we can just say maybe F2. The reason why we are saying these are F2 is because this is the parental generation, then first filial generation, then the children of these are considered to be second filial generation. So this is how we can answer a question like this one. So that's it for this video. Thanks for watching.